yesterday we looked at, we finished off here how we saw that sickle cell anemia or the sickle cell trait, we can see both complete dominance and incomplete dominance in terms of the shape, okay, of the cells, the red blood cells. So in sickle cell trait, remember that in normal oxygen, the shape is the donut shape, okay? Well, if the cells are exposed to prolonged exposure to low oxygen levels, you get the sickling. Some cells sickle, so it's incomplete dominance. But in terms of the amount of the hemoglobin, we find that both HbA and HbS, so we find it 50-50 ratio. So what's that? What sort of
codominance?
Codominance, okay? So that's codominance. But in terms of the shape, we see complete dominance.
But when
in normal oxygen, okay?
Yeah,
But when exposed to low oxygen, some cells become sickle, we see incomplete dominance, okay?
Yes.
So three different types of phenotypes we see here.
so we call it codominance.
Yeah, so at the protein level, it's codominant, okay? But at the shape level, we see two sort of dominance. We see incomplete and complete dominance. So we finished off here. I think we finished on the last slide there. So today we'll continue with the different types of non-Mendelian genetics. So in polygenic traits, the trait is controlled by more than one gene. So poly means many, so it involves many genes. And examples of this are like eye color, skin color. So the accumulated expression of many of these genes can determine the color of the eye. So you get from one, for example, from blue to black, okay? You get a variation in between depending on the expression of the different genes. So you get a continuous variation. And this sort of eye color, skin color is dependent on melanin deposition. Remember last week we talked about the albino trait? That's, yeah, normal skin color in albinism. That's controlled by
Melanin.
one gene, okay? So whether it's complete dominance, okay? In terms of normal skin color to albinism. Albinism is recessive, okay? So here we're talking about the levels of melanin rather than absence or presence of it, okay? So many genes control the levels of it, and depending on how much it's deposited, you can get a variation from extreme dark color to light color pale skins, okay? Depending on how much melanin is in the skin. And this is obviously controlled by polygenic inheritance. Many genes are involved, okay? So other types, other traits that are controlled, like height, body size, body shape, these sort of characteristics are also polygenic. And for all of the genes involved in polygenic traits, you find an allelic pair. So remember that you get an allelic pair. Remember, one is on each homologous chromosome, so one of the alleles for each gene. So one from the mother, one from the father. Yeah. So, for example, if eye color was controlled by 3 different genes, say we denote it as A, B, C. So there will be an allelic pair for each of those genes. Okay. So, for example, if they're homozygous dominant, that all of them will be capital, huh? So in polygenic traits, all of the genes come in allelic pairs, okay?
Okay.
So you get this quantitative effect, okay, from the dominant one, all right? The dominant trait is only appears if it's homozygous, okay?
What does the quantitative effect mean?
Quantitative mean it's like the additive effect of all the different alleles, okay?
Yeah.
Gives the phenotype. So what we see is the homozygous for dark color, for dark color, for the extreme dark color, it will be homozygous, okay?
Okay.
And also for the pale color, homozygous. But when you cross these two, okay, you don't get dark color or pale color. You get in between, in the middle. So the heterozygous shows incomplete dominance. Remember, a mixture of the two.
Okay.
So the heterozygous shows incomplete dominance. If you have a look at this, for example, this shows, for example, for height, okay? So you will have, homozygous dominant will be for, so you need the homozygous dominant alleles, okay, to be very tall, okay? To be short, it's homozygous recessive, okay? But in the middle, when you cross these two, the heterozygous, okay? The heterozygote is neither tall nor short, so it's medium height, and this is where we see incomplete.
Okay. So homozygous can be dominant and heterozygous incomplete dominance.
Yeah. So the heterozygous, okay?
So are all polygenic inheritance incomplete dominance?
Yeah. The heterozygous, yes.
Of all of them?
Yeah. Because it's a variation, it's a quantitative effect. So, for example, in height, in eye color, in skin color. So,
Doctor, all of
it's
them
all, all of them are a continuous range, okay? So you will see in these sort of polygenic traits, the heterozygous or the heterozygotes, which it has one of the dominant, one of the recessive alleles, is shows the phenotype in a complete dominance, because in this case, you see, if you have the heterozygous, it's neither tall nor short, it's medium, okay? In eye color, the heterozygous will be in between dark and blue color, okay? Somewhere in the middle, probably, I don't know, green or something like that, okay? So,
all of the polygenetics have three alleles.
no, this is just an example, okay? We're just saying if it was, it's probably controlled by more than that.
Correct.
Yeah, I don't know the exact number, but each one of these is controlled by more than 3 genes.
Oh,
Okay, it's a continuous variation of all those genes.
okay.
So it's not 3. Okay, this is just to show you an example to make it look easy. So we see that as a continuous range. Okay, you see it in the eye color, in the height, and also in the skin color here as well. All right, and the trait that you see in between the two, the heterozygous, is showing incomplete dominance, okay? So most of the people we see here are in the middle. There's a small percentage, it follows that normal distribution, in maths, normal distribution.
Yeah.
So most of the people are in the medium height. There's very few people who are very tall and very few people that are very short, okay? Or with the eye color, the same thing, or skin color, okay? So just to recap, okay. As I said, they behave in incomplete dominant manner in polygenic traits, okay. The heterozygous, okay. Heterozygous because the homozygous dominant is very tall, homozygous recessive is short, and the heterozygote is medium, so it's in between. Now, what you do is if you cross, remember Mendel's
Yeah.
cross?
Yes.
If you do a cross
Dihybrid.
between them, dihybrid. So this is not a dihybrid cross. It
Or
will be
triple hybrid.
polygenic cross or whatever, polyhybrid cross or whatever you want to call it. So you will see a mix, okay? A mix of the tall and short alleles, okay? So you will see a few, maybe very few with the tall phenotype and very few with the short. Most will be in the medium, okay? Or it will be like a continuous.
That's right.
Yeah. So if you remember the dihybrid cross, how we got only one of them,
Yeah.
93, one of them ended up.
Homozygous
yeah.
recessive.
So there's the chance of that very
Weak.
weak, okay?
Yeah.
And remember we talked about the environment as well affecting the genotype.
Yes.
It's not just the genotype. Remember in the case we just discussed about sickle cell anemia
Yeah.
at low oxygen,
Yeah. It's incomplete dominance.
incomplete dominance. So
Yeah.
the genotype is not always involved in it. So the environment is having an effect. In this case,
Yeah,
the oxygen
oxygen.
is affecting the
Yeah.
phenotype. So normal oxygen, it's complete dominance. If we change the environment, it becomes incomplete. So you see the environment has effect. Another example is, as I mentioned before, if you're very tall, you're living in a country where there's nutrition is very low, you're not going to be very tall, okay? If you have the genotypes for being very tall, okay?
That's why in Tokyo everyone is short.
Is it?
In Japan, everyone is short. They're so
But-
short.
But they have a good diet.
Everyone is short.
In South Africa, they're all taller.
Yeah.
Don't have nutrition.
But what if they had the good nutrition? They might be even taller with the genetics.
Yeah. Like
Yeah.
the American Africans, they are more taller than the
Yeah.
Africans.
Yeah.
That's
So-
because they have more nutrients in America.
Another example is lactose intolerance. The mutation that causes people to have lactose intolerance is they are unable to digest galactose. Remember, it's part of the
Yeah.
lactose is made up of sucrose and
and glucose.
sucrose,
No. Sucrose.
galactose.
Yeah, galactose.
Sucrose and galactose.
Galactose, yeah.
So people who are deficient in this enzyme, unable to digest lactose, is very, harmful to them if they eat it. But so this is the environment. So the food is having the effect on the genotype. But if you remove the food, you're normal.
Mhm.
So the phenotype is not showing. Okay.
Yeah.
So another case where the environment is affecting the genotype. So no, if you don't eat lactose, you don't show the phenotype,
Yeah.
lactose intolerance. If you eat it, you will show it. So the environment is affecting here again. And these are some of the examples, all right? So nutrition affects body height and size, okay, as we mentioned. Another example was that galactose. Another example is the sickle cell anemia, where the oxygen levels is affecting the phenotype. So we can list many more, but that's gives the idea. All right. Let's move on to some chromosomal disorders. So we talked about how you get in meiosis. Remember, in meiosis 1, homologous chromosomes separate, and in meiosis 2, sister chromatids separate. But sometimes they fail to separate, and we call this non-disjunction of the chromosomes or failure of the chromosomes to separate. It can happen during meiosis 1 or meiosis 2. So here is the normal meiosis, remember the homologous chromosomes separate in meiosis 1, then you get separation of the sister chromatids, okay? Now, there's two ways to get non-disjunction, failure of the chromosomes to separate. And this might happen to an individual chromosome. It not necessarily happens to all of them, okay? So it can happen to one or two, whatever, all right?
Okay.
Not all of them. So we're talking about a pair of chromosomes. The rest might separate properly, okay?
Okay.
So, for example, in meiosis 1, when you get non-disjunction, remember the failure of the homologous chromosomes. So if this has 23 pairs, okay? One of the pairs didn't separate, so this ended up with how many pairs? So it ended up with
46. 22. 22 pairs.
24, 24 chromosomes.
Yes. 24. Yeah, 24 chromosomes.
24 chromosomes, huh?
Yeah.
24
Chromosomes.
duplicated chromosomes, okay?
Yes.
So it will end up with 24 duplicated chromosomes.
Homologous
Correct?
chromosomes.
Everybody agrees with that?
How it can increase to 23 to 24?
So it's 23 pairs, okay? We separate homologous chromosomes. So you end up with here sister chromatids, okay? 23 sister chromatids. But we also ended up, because one of the chromosomes didn't separate, it came along with it as the homologous chromosome.
Yeah.
We got non-disjunction, okay?
So there's 24.
So now there's 24. Here we got
22.
22 duplicated chromosomes. Or sister chromatids, in the form of sister chromatids, alright? So everybody's following me?
Is it homologous in meiosis 1?
Yeah. So the homologs failed to separate.
Yes.
So you ended up with a
24.
chromosome here, one of the chromosomes, okay?
Oh, okay.
Okay? So then, when you have meiosis 2, what happens here is you ended up with 22 chromosomes, okay? Here, 22 here,
Unduplicated.
here, what did you end up with?
24.
24, 24,
Unduplicated.
unduplicated, okay? Because meiosis 2 separates what?
Sister chromatids.
Sister chromatids, okay? So this is one way of getting a non-disjunction of chromosomes.
So just
This can happen in the autosomes or the sex chromosomes, okay?
Okay.
Yes?
just to make sure, we have on the left side we have 23 duplicated homologous chromosomes and one non-homologous on the left side, the 24 duplicated.
So here on the left, you're saying there's 24, huh?
There's 24.
Yes.
So,
So normally we have 23 duplicated chromosomes, okay?
yes. Yeah.
But in this case, one of the chromosome pairs failed to separate. We got non-disjunction. So it carried an extra chromosome with it, didn't it?
Yes. Yeah.
So it ended up with 24 duplicated chromosomes. In this cell, while this one is now missing one, it's
Missing one,
22, okay?
Yes.
So this can happen both in the autosomes and the sex chromosomes, all right?
Okay.
So we ended up with 24 unduplicated in these cells and 22. They're missing one of the chromosomes, okay? Now, we can have this happen in meiosis 2, non-disjunction. So you get normal separation of the homologous chromosomes, okay? So you end up with 23 duplicated and 23 duplicated. So one of the cells, separates normally. So you get the separation of the the sister chromatids, okay? So you get normal, what do you get?
Unduplicated.
23 unduplicated. Okay? So what happens is that in this cell, you get non-disjunction of one of the chromosomes, okay? So we have 23, but if you get non-disjunction,
There.
you end up with,
Duplicated.
so you end up with how many?
23
23
duplicated.
duplicated.
Yeah.
No, 20,
24.
Oh, 24.
24 unduplicated, and in here 22.
Oh, yeah.
Okay,
How?
because this one chromosome failed to separate. Now you got
24 chromosomes.
24 chromosomes. The
Unduplicated.
22,
Unduplicated.
the other ones separate. We're talking about a single one doesn't separate, okay?
Okay.
So you end up with 24, similar to those ones, okay? Unduplicated.
Okay.
One of them is duplicated, okay? But
So it will be
it'll be 24. It will act like 24 chromosomes, okay?
23 double.
If it's fertilized, okay?
But Doctor, 23 unduplicated and one of them duplicated, right?
So, well, normally you should end up with 24 or 23 here, because
Yes.
one of them failed to separate. So it should be 24.
All of them unduplicated. Well, one of them failed.
Yeah, yeah. One of them is duplicated, but it'll act as it's, once it's fertilized, it'll act like an independent chromosome. Okay.
Now what we call 23 unduplicated.
Yeah. So 23 unduplicated or non-sister. One of them is the sister chromatid failed to separate. Okay. But you got 24 chromosomes in the end, the total, all right?
Yes.
Okay. And this one has 22. And this is important for some of the disorders that we talk about, like Down syndrome, Turner syndrome, Klinefelter syndrome, where
Trisomy.
these have abnormalities in the chromosome number. So, is everybody okay with chromosome non-disjunction happening in meiosis 1 and how it's happening in meiosis 2?
Yeah.
Okay. I think we covered that.
Yeah, okay.
Yeah. I just covered it in that time. There's nothing here that we don't need to know. So basically, to recap, so non-disjunction, failure of chromosomes to separate can happen in the autosomes or sex chromosomes, and so you can so the non-disjunction changes the chromosome number in the sperm and egg. You can end up with 2 copies or no of a particular chromosome, either with 2 copies or instead of 1, you can end up with 2 or none.
Okay.
So it says here 2 copies, but also you can end up with
None.
0.
Okay.
Because we can see here these cells didn't get one of the chromosomes. So for that particular chromosome, they get
0.
and that's important for some of the diseases. So if you end up with 3 chromosomes for a particular after fertilization, so if you have a non-disjunction in one of the chromosomes, separation, and you end up with 24, and that 24 is fertilized. So 24 plus 23, what does it give you? 47, which is 23 pairs, 22 pairs, sorry, and 1 triplet. Correct?
Yes.
So we call this trisomy, and this happens in Down syndrome. It happens in so trisomy 21. You got trisomy 18, trisomy 13. So different diseases involved in trisomy, but a very characteristic one that probably seen people with this is trisomy 21, Down syndrome.
Yes.
You might have seen people with that. So the features, we'll talk about it next. So in trisomy, you get 22 pairs, and one of them is in triplet. Okay, and this happens because it can happen by non-disjunction in meiosis 1 or meiosis 2. Okay? So you can, if you fertilize this one with a normal sperm, say it's an egg, you'll get trisomy 21. If you fertilize this one with a normal sperm, you will also get trisomy 21. Okay? If that is the failure of chromosome 21 to separate, alright? So in both meiosis 1 and meiosis 2. If you have only one of the homologous pairs for one of the chromosomes, it's called a monosomy. So you have a cell, a gamete with 22, and you fertilize it with a normal sperm, okay? So an egg, what you end up with is 45, so you end up with 22 pairs and one single chromosome. Alright? Or you can call it the same one. So,
I forgot the 5. 4 or 5.
nice. There you go. So, monosomy, and you get this in Turner syndrome, okay? Where they have missing one of the chromosomes, okay? So these are females, they're missing one of the chromosomes. They can be fertilized with, you know, a sperm coming from here, okay? So if it's a female, if it's a normal egg, okay, 23 chromosomes. Now, can it happen in the sperm and in the egg for that one? Because it's a sex chromosome that is
Single.
altered. Yeah. So if you had,
It can be.
if you had, you know, the egg is always X, remember?
Yeah.
So for an egg without the X chromosome, so if you have a non-disjunction in the egg, okay, so it'll end up with 22 chromosomes minus the X chromosome. So it has to be fertilized by what? A sperm that contains X, okay?
If it was Y.
If it's Y, it's not going to be Turner syndrome. Because,
Oh, yes.
yeah. You understand that? So, if it was if the egg was normal, so you must have the sperm without the sex chromosome to fertilize it to end up with the same disorder. Okay? Got that?
Yeah.
So it's X0, that means it's missing one of the
Yeah.
sex chromosome. If the egg is normal, it's got the X. The sperm must be missing the
6 chromosomes.
sex chromosome that fertilizes it.
The sperm is missing.
Yes. So a sperm, one of these sperm will fertilize it, containing 22 autosomes and no sex chromosome, okay? So a
Is
failure,
there any human with no sex chromosome?
well, I mean,
And he will die.
there's a lot of important genes. You only get, you get these sort of variations, there's other variations of this X, Y, Y, where you get failure of the,
XYY, he can live.
yeah, X, X, this is a female, okay? But she's missing, okay?
Could she live without the other chromosome?
Yeah. These people can live, it's okay. Alright, so that's monosomy, when you get 22 pairs and one of the chromosomes is a singlet.
Yeah.
So as we discussed, Down syndrome is usually known as trisomy 21, okay? Because there's a failure of there's a non-disjunction of chromosome 21, which leads to trisomy 21, okay? So you have 3 copies of trisomy for chromosome 21. Now, this risk for this increases in females as they get older, so the older age, over 35, the risk increases for non-disjunction of chromosome 21. Is this inherited?
No.
Maybe.
No.
It's in the sex.
Could be.
But it's not inherited from the parents. There's a failure of the chromosome. They had the normal chromosome, just they failed to separate.
Yeah.
Yeah. So it's not an inherited disorder. It's a failure of the chromosome, and it increases with age. So the female, if they're over 35, they have more risk of having a child with trisomy 21. So from that we understand that the non-disjunction is happening in the egg, huh? They actually have some tests they can do to identify the genetic test. So they do a chromosome, you know the karyotype?
Yes.
They can do a karyotype on the unborn fetus. They just take some fluid from the amniotic sac,
So
and then they
can
test
avoid
it.
that.
Well, they can terminate the pregnancy,
Yeah.
abortion, early stage, you know, before 21 weeks or I don't know, something like that. So there is some testing of the fetus that can be done to detect this at early stage. So they take some fluid from the amniotic sac and tested the chromosomes to see if you have.
Can they treat him?
No, there's no treatment.
So they will
why
kill
they
him.
detect it?
If you're not gonna have an abortion, there's no need to do the test because the test itself increases the risk of abortion. If they don't have there's a risk with the test that the baby can
Oh,
be terminated as well. So you have the option to do the test. I think they also have some blood parameters that show some risk.
is it halal?
Well, of course it's not. We don't have abortion. Do we have? Only in cases where you got what we call ectopic pregnancy, where the mother is at risk, where the actual the embryo attaches into the fallopian tube and it grows and it's very painful. If you don't remove it, it will kill the mother.
Yeah.
Yeah. So you must those sort of pregnancy. I think we're allowed to do that. I mean that's the saving a life.
Yeah.
And I think at that stage the ruh is not entered
No,
the,
it's
Yeah.
No.
After 3 months,
months.
Yeah.
I guess.
so it's not considered living at that stage, is it? I don't know. I'm not a religious scholar,
Yeah.
so I don't know about the ethics of it, okay? Islamic ethics. Maybe they should put that in the subject in Islamic ethics. They should be teaching you that. You're going to medical school. Do they teach any of these ethics?
I guess later.
They don't?
I don't know, I guess later more.
Not like.
What about
in terms
medical?
Of yeah,
They
not
should do in
medical.
terms of medicine, I think. They should change it because you need to know these. Alright, so characteristics of these sort of people, they got this short, round face, you know, and you see all of them have this sort of feature. The people
Yeah,
with-
look alike.
yeah, they're not tall, they're very short. They have breathing difficulty, problems with this respiratory, they have heart problems. They have slow mental development, okay? And they have shorter lives than normal people.
The
Because of
maximum is 25, I guess.
difficulties in the respiratory system and in the cardiac system, okay? So they tend to have shorter. But, you know, with some education and, learning skills, these people can have normal lives and, contribute to society and
But their normal life is 25, I guess. Something like that.
I think they can live a bit longer. I think it's 40 years.
Yeah. I know a person who's like 36.
No, I guess
Yeah.
I mean the
Yeah.
normal.
Yeah. Well, I think the average, they can live up to 40.
But they're like smart in certain areas. Like
Yeah.
they can memorize by reading once or like by
Maybe they have some.
they have some abilities that like
People with
nobody-
schizophrenia have some ability to also, they're very smart, but
Yeah.
Allah يخفي عليك.
yeah. So it's yeah. They're mentally retarded, they have heart problems, respiratory problems, and usually death at young age, okay, because of this extra chromosome that they have 21. Also, they show they're very happy, cheerful, affectionate as well. You
Yeah.
see them, okay? That's it, which is a good thing. I think we should all be like that.
Yeah.
We don't need to try number 21, but
Yeah.
that's something that we can do with a bit more. All right, so some of the other alterations in the chromosome number we see in the sex chromosomes. So trisomy 21 is a autosome disorder, okay? Alteration in the autosome. And these Klinefelter and Turner syndrome, these are alteration in the sex chromosomes, okay? So normally, female is lacking the Y chromosome, correct? Yes? So a female lacks the Y chromosome, huh?
Yeah.
No.
Yes, they have 2x. How?
Amen.
Yes. That's the normal chromosomes of a female is XX, huh? So to be male, you must have XY. Female, XX. Okay. So in these syndromes, for example, in the Klinefelter, you have XXY.
He's
Now,
between.
yeah. Well, it's a male. Okay. With sometimes features of the female, like enlarged breasts. Okay. They might have some problems with production of the male the testosterone. Okay.
Yeah.
They can be sterile, but with new technologies, you know, assisted fertilization, these people can have children. Okay. They're not always sterile. So the features are, you know, they're males. So because of they got the Y chromosome. Now, how can that happen? There's two ways it can happen. What sort of non-disjunction can cause that?
The egg will not separate.
So in the egg, the XX didn't separate, correct?
Yeah.
What about in the male, the sperm?
It's a
Failure of the XY to separate.
Yeah, both of them.
Oh, okay.
So you can see you can get it in two ways. So if you have an egg with XX that is fertilized by a Y sperm, or if you have an egg with normal X and it's fertilized by XY sperm.
Yeah.
So two ways to get that. So non-disjunction happening in the egg or non-disjunction happening in the sperm. And this can happen both in meiosis 1 or meiosis 2, okay?
Yeah.
So these people, they have like sometimes enlarged breasts, which they can be corrected with surgery, okay? So, and because they have the extra X, sometimes they're also slightly more prone to breast cancer than normal males.
They can choose if they want to be a male or female.
Well, they're male.
No. Then when they
Have all the
bigger,
reproductive parts. It's just the imbalance in the
if
hormones
he
that
feels
causing.
he's a woman, he can
Well,
translate.
this appearance doesn't come.
In Islam, he can choose.
Yeah. Later on, okay.
Yeah, later on.
Yeah, the puberty onset will show some of these symptoms. Not it's not detected before puberty.
Yeah.
So the other one is the Turner syndrome, and these are female, obviously, because they have one X, so they're female. They're missing one of the X chromosomes. So we said this can happen in. So, for example, a female with no sex chromosomes, an egg with no sex chromosomes, okay? So no sex chromosomes, it's fertilized by a sperm with eggs, okay? Or a sperm without any sex chromosome can fertilize that and give you eggs, okay?
Is there sperms without any sex chromosomes?
Yeah, it can be
Non-disjunction.
non-disjunction
Okay.
of the sex chromosome. So this condition is rare, okay? these
mostly
people are,
about
these are very short people, okay? Most of the embryos with this genotype, so the XO, are terminated early on, okay, by themselves, all right, because they have lots of problems, you know. They're short, they're sterile, normal intelligent, but they have small breasts, okay, and they usually have this webbed neck. You can see a web on it, short. The back is a bit bent, and they're sterile, but
What's sterile?
sterile means they can't have children.
Okay.
But they're, with assisted technologies like fertilization, they might be able to have children. And
Can't.
there is, you know, here we're showing just where the other X is missing, but there is variations in this disorder. Sometimes parts of the second X chromosomes are there. Depending on which parts, the severity of the syndrome can vary, okay? So not always you're missing the X chromosome, okay? There is variations to this disorder, but in terms of the chromosome number, this is where we're talking about alterations in chromosome number. The XO is related to that, okay? But there is variations in this disorder where parts of the X chromosome are still transferred to the egg, for example, from the sperm. Not all of it is missing. As we talk here, in the next slide, you'll understand that when we're talking about changes in the chromosome structure, okay? So changes in chromosome structure can also cause Turner syndrome, okay? For example, changes in the X chromosome, parts of it can get deleted, all right? Or translocated or whatever, all right? So Turner syndrome, you got one X female, they're short, sterile, normal intelligent, but smaller breasts, okay? So alterations in the chromosome structure can also cause diseases. So you can have a deletion in the chromosome, okay? For example, if you take this chromosome and one part of it is deleted, so now you have a chromosome with missing DNA.
These are alleles.
No, these are just the chromosomes in made into sections for you to visualize the deletion, okay? So these are sections of the chromosome, okay?
Is mutation?
So for example, a mutation, of course it's a mutation because it's a deletion. A whole chunk of the chromosome is deleted and removed, okay? And now it's missing that part, okay? It might contain vital genes, okay? So now the chromosome that has the deleted is missing that DNA and also missing the genes. And very normally, because they're missing the genes, all of these deletions are aborted, okay? Early on. So they're fatal, okay? That means lethal means that they're fatal to the embryo and the embryo does not continue because it's missing vital genes. Sometimes you get a viable embryo and it continues to form and you get a child. For example, in this disease, Cry-do-chat disease, this syndrome, there's a deletion in chromosome number 5, okay? So, but the people who have this, they're very mentally retarded. They have this kitten cry. They cry like a kitten because of the problem with the small larynx, okay?
Yeah.
So in Cry-do-chat syndrome, there's a deletion in chromosome number 5, which leads to very mental retardation, this characteristic kitten cry because of the small larynx, and that's how you can detect this disorder because of the deletion in chromosome
But they
number
are
5.
normal in general.
No,
they
they have
don't have any symptoms.
mentally retarded.
Yeah.
This is there's a severe effect on it because there's a deletion in chromosome number 5.
So you can detect them if they cry, only if they cry, or like by
Usually
their
this is a characteristic of this disorder, but you know, the physician will order probably a to confirm it. I mean, they can check at the anatomy of the larynx, you know.
But if
If they're not sure, they can do a karyotype and detect it. You can see a change in the structure of chromosome number 5.
yeah, but like if
Because
they
of
have
the
small
deletion.
larynx, they will have like a small voice.
Yeah.
Like,
Yeah, well, to really to confirm it, you would need to look at the structure of chromosome number 5 in the karyotype. Remember, that banding pattern changes. The banding pattern will change for chromosome number 5 if there's a deletion. Okay.
Yes.
So another type of structural change in the chromosome is called translocation. It can happen between non-homologous chromosomes. Okay. So in this first case, you see here, what's happened here is that you have an exchange of, so you got A, B, C, D, E, F, G, then you got H, I, J, K. So what happens is we got this reciprocal translocation. So a part of chromosome, this non-homologous chromosome KLMN has translocated to the, so let's just label it chromosome A and chromosome B, okay? So this is the new chromosome A,
A prime.
and this is the new chromosome B to make it. Alright? So what happens here is that there's a what we call a reciprocal. So a part of B is translocated to A, and a part of A translocated to B. This is called a reciprocal translocation. So you see here, if you have a look at that, from here, there's the translocation. So N has translocated to chromosome A, and HIJ has translocated to chromosome B. So basically what happens is that a piece of it is excised, you get breakage in the DNA, double stranded break, and a double stranded break in this one, and the two pieces translocate to one another and there's joined to each other. Okay? So this is called a translocation. And this can affect the expression of genes. What if it happens in the middle of a gene, okay? Disrupts the whole gene. Or it moves it to another chromosome where that chromosome is not expressed as well. Expression is changed, okay? This can increase the risk of like leukemia, okay? And like certain cancers, like leukemia. So some of the leukemias, they're because of these translocations in the chromosomes. And it changes important genes, maybe cell cycle genes, okay? That can affect, that can cause cancer. So translocation, if it happens, for example, in the middle of the gene, okay? Now, part of that gene is on a different chromosome. It can't be expressed properly, okay? Or on the other chromosome, the expression changes. It's low expression or high expression. And this can cause disorders like leukemia, cancer. Okay? So we understand deletions and translocations?
Yes.
So translocation can happen, so reciprocal means it's happening on a different, it can also happen on the same, okay, chromosome. So, you know, part of it can break off and go, you know, instead of being here, it will end up here, okay?
What do you mean?
They can switch, you know.
Ah.
So that is the side.
On this, so it can happen on the same chromosome or on a different chromosome, okay? All right, let's have a look at this activity. How much time do you have?
5 minutes.
We're over time. Nobody's telling me.
It's okay, 5 minutes we have.
I have to go to the other class, but we'll finish this. Next week we will organize a probably not next week, the week after, when before the exam, biology exam, hey? And refresh the revision, okay?
Alright.
Okay.
Who's the group leader? You're the group leader?
No.
Can you send me on WhatsApp so we, you know, okay? All right, so let's answer these questions quickly, then we can go. So in Mandela's experiment, crossing a yellow and green pea plant, okay, the first generation showed yellow phenotype, while the second generation showed the yellow and green phenotype with a ratio of 3:1. Why a green pea skipped the generation?
Because it's autistic.
Yes.
B.
Yeah, B.
Yep.
B.
Okay. What type of dominance is shown here? So we got a red or a pink flower and a white flower. You get co-dominance, huh?
C.
What's shown here? You get a red
Poly.
and a white flower. You get a pink.
Incomplete.
Incomplete. You cross a red and a white flower, and most of them are red.
Heterozygous.
Complete dominance.
B, yeah.
And you get a variation here.
Polygenic inheritance.
Polygenic, okay? Okay. So we got that?
Yes.
Okay. So send me a WhatsApp and next week, inshallah.
have you-
Not
I will
next
send
week,
it
the
to
week
you.
after.
I will send it to you.
We'll do some revision.
Inshallah. The last one was what?
Polygenic.
Polygenic.